Hi guys, in this video we're going to be talking about genetic engineering. This is going to be a bit of an overview because uh, the contents covered in genetic engineering are quite broad and bring quite a few things together. So in this initial video I'm just going to kind of do an overview of the topic just so that we can understand what the whole purpose of genetic engineering is and what are the key steps in the process. And what we'll do then in subsequent videos, what I'll be looking into is how a closer look at how those steps are carried out. So let's begin. So the whole idea behind genetic engineering is a manipulation of the gene expression process. So these are cells, so let's just pick one cell out. So this will be our organism A. Okay, so this could be one cell of organism A, and that organism could be anything. Okay, and we'll look at a few examples as well later on. So this is a cell of an organism. It has a genome. Okay, so I've drawn the genome circular here, but if it's a bacteria, it's going to be a circular genome. If it's an animal cell, it's going to be linear with chromosomes, etc. But just for simplicity, we're just going to be dealing with this. So let's say organism A has a certain number of genes in its genome. So it has the gene for protein A, it has gene B, C, D, E, F, and G. All right, so all these could be different genes expressed by this organism A. But what does that mean? That means that this organism has the capability to transcribe these genes into mRNA. So, for example, gene D might be transcribed by this organism. So the DNA is transcribed into mRNA. And the mRNA is then read by ribosomes, codon by codon, and together with the tRNAs, the amino acids are brought together in a line, in a sequence, joined together with condensation reactions forming peptide bonds to make a polypeptide, which goes through the different levels of folding, secondary structure, tertiary structure, quaternary structure, to become a functional protein. Okay, and this is the key, that this protein therefore endows the cell, the organism, with a particular function. Okay, so that might be an enzyme, in which case it's going to catalyze a reaction. It might be an antibody, it might be a protein in the membrane that acts as a channel. Whatever it is, its function is due to the cell being able to express that protein. So, in terms of language, we're going to be talking a lot about expression. So let's just define what gene expression is. Gene expression is for a gene in the genome to be transcribed and then translated into a product that has a function. Okay, that is what we're going to be calling gene expression. Right, so we've looked at the simple example of organism A being able to express one of its genes, gene D, into a functional protein. We could have another organism B, different species, and so we must assume that as well as having some common genes, as all organisms do, it's got some different genes that give it different characteristics and different functions. Okay, so we can have another organism here with its own genome. Okay, so we can say that, right, maybe it has some genes in common, but then other ones are different. E, F, G, H, I, J, K, L, M. So organism B has some common genes with organism A, but it has a lot of other genes which are not common to both organisms. So what is genetic? So what we must have the idea in our minds is that organism B is also transcribing and translating its own genes to make functional protein products that endow organism B with different characteristics and different functions. So, what is genetic engineering? Genetic engineering is a manipulation of this system so that you can allow organism B to express a gene from organism A, a gene that you know of. You want organism B to be able to produce a functional product from a gene that is in organism A. So essentially, you can think of it as a physical extraction of a gene from organism A, inserting that gene into organism B, and then giving organism B the chance to then produce protein product for which it didn't originally have the gene for. Okay, so what does that mean in our example? So for example, we could say that we want organism B to produce a protein product from gene E. Now gene E, hope you can see, is not present in organism B. So what we can do is we can extract E from here, so we can extract it. So what I'll do is I'll give this genome a black color. Okay. This genome can have a white color. So what we're saying is we can extract the gene from organism A and then insert it into organism B, and by doing that we produce technically a new organism which is called transgenic. So we produce transgenic organism here. So this is a transgenic organism because it has its original complement of genes as well as a gene from a different species, a different organism. Okay, so the idea of a gene from a different species is the key here. Okay, so what we'll have is a new genome looking slightly different. Okay, so we'll have A, B, C, H, I, J, K, L, and M. And somewhere in there, as well as all the original genes, there'll be the gene that we introduced from organism A. So now organism B is expressing a gene from organism A. It has the ability to produce this protein product that it didn't have before, and so it's got the ability to do something new or produce something new. Okay? All right. So as well as all the other genes, it's now got E as well, and that makes this a transgenic organism. So that's very simply what genetic engineering is. It's taking a gene from one species, inserting it into another, allowing that organism to be then transgenic and produce a protein product for which it did not originally have the gene, but now it does. Okay, I hope that's making sense. So what we'll do now is we'll look at some examples of why you would want to do this. Okay, so for example, okay, so I've just modified my diagram a little bit, just annotated the key steps. Okay, so now we'll begin, right? So what I would like you to consider now, there's two main examples that can illustrate this idea, okay? And just like we said, you know, I've kind of used a bacterial example here just to keep things simple, but you know, the two different species of organisms can literally be anything, okay? So our first example is that of insulin. Okay, so there are disadvantages with having to extract insulin from animal sources, so you should know those. But because of those disadvantages, you could genetically engineer organisms that don't normally produce insulin to be able to produce insulin and therefore get a different source, an alternative to extracting insulin from animals, for example. Okay, so, and that will have its own benefits, but that's not for me to look at right now. I'm concerned with the mechanism of how this is working. So in terms of insulin, organism A would be, for example, a human, right? Because we want human insulin because it is to treat a human disorder. So organism A in that case would be human. Okay, and specifically we would want cells, we're more likely to find this gene expressed in the cells of the pancreas. So that's where we would look first, but perhaps I'll come back to that at a later stage. So we would want human cells which would have the gene for human insulin. So human cells expressing human insulin gene. Okay, remember there's a gene for insulin in the DNA of us and in our DNA, and that gets transcribed into mRNA, and the ribosomes put the amino acids together to make it into the functional insulin hormone. Okay, it is a peptide hormone. So that's the situation in organism A, but it's not practical to extract insulin from human beings. So what we'd rather do is put it in bacteria, which multiply really fast and can produce really large quantities of insulin, and much more cheaply than extracting it from pigs. So, as well as other advantages. So let's do that. So what we would do is we want bacteria to express this gene so we can extract human insulin from bacteria. But problem is, bacteria don't express human insulin. They don't have the gene for human insulin, obviously. Okay, so what we do is we take the gene for insulin, we take our gene, cut it out of the DNA here, and so we get some human cells first of all, and we cut out the gene for insulin, and we transfer it into a bacterial cell. We insert it into the genome inside a bacterial cell, and we produce this transgenic bacterial cell that, so in this case, in our example here, our organism B would be bacterial. A bacterial cell, okay? Right? Obviously different species, right? Human cells, gene from human cell going into the bacterial cell. So once that gene is in the bacterial cell, our bacterial cell is now transgenic, because as well as all its genes, it's now got the human gene for insulin, and it will start expressing that to very high levels, okay? As long as you put the correct kind of regulatory bits on there, okay? So now the bacteria is producing human insulin. So the insulin gene is there. The gene is being transcribed into mRNA by the bacterial RNA polymerase. That mRNA is then floating around in the cytoplasm, locks onto a bacterial ribosome, and then the ribosomes put the amino acids together according to the instructions in the mRNA, and they produce a human insulin peptide. Yeah? And then what you can do is then you can extract the insulin from the bacterial cells and use it, right? Okay? Didn't have to come from an animal. We could just do it like this, okay? So, key points we should note here are bacterial cell, transgenic organism, produced, so transgenic bacteria now express human insulin gene, and in very basic terms that means human insulin is produced inside the bacterial cells. Okay, so this is just another way, less scientific way to say that. Okay, so this term expression is going to come really handy. Do use it, do understand what it means. Okay, so that's our example of the insulin. But there are other examples as well. Perhaps I'll use a different color for this. Okay, so the other example is, so that's example 1. Example 2 is the golden rice example. Right? So in the golden rice example, the organism A, i.e. the organisms that have the genes that we want, are the daffodil plant, okay? Daffodil plant, and certain bacteria this time that express certain enzymes. With the golden rice example, you have rice which has normally very low levels of beta-carotene. Now, beta-carotene is something that's lacking in diets of certain parts of the world population. So one idea was that you can increase the level of beta-carotene in rice, which was consumed quite a lot by that population. So one way to get more beta-carotene in the diets of this population would be to increase the level of beta-carotene in rice. So how do we do that? So it's important to note that in the rice plant, the starting material needed to produce beta-carotene is there. It just doesn't have the enzymes to convert the starting material, the starting compound, into beta-carotene. So the idea was that you insert the gene for these enzymes into the plant, producing a transgenic rice plant that would then have the enzymes to convert this starting compound into beta-carotene at the end. So our organism B then would be the rice plant. Okay, so in our golden rice example, organism B, i.e. the recipient organism, in a way, that's not a technical term, I think, okay, is the rice plant. So the rice plant then, it needs certain genes to convert this compound into beta-carotene, and the things that it doesn't have, the enzymes that it doesn't have, were found in 2 different organisms. So daffodil has an enzyme called phytoene synthetase, okay, and bacteria have an enzyme called CRT1. Is it really necessary that you know exactly what these things are and what they do? Not really. I think at this stage there's more value in just understanding that there's something the rice plant needs from the point of view of the human, and the genes that it needs are present in these other organisms, and we'll be transferring them from one organism to the other. Okay, so daffodil and bacteria then are our organism A, and what we can do is we can extract the gene for phytoene synthetase and put it into the genome of a rice plant cell, and we can extract the gene for CRT1 and also put it into the rice plant cell, producing a transgenic rice plant which now has 2 extra genes, doesn't it? Yeah. Okay. So yeah, let's just call it E and F. Yeah. So now the rice plant has got 2 genes. It's now expressing, the rice plant, our transgenic organism, is now expressing the enzymes needed. It's expressing now phytoene synthetase, it's expressing now CRT1 protein. So now it has the enzymes to convert what it's already got, this starting compound, into beta-carotene, and lo and behold, we have rice now that has high levels of beta-carotene, or at least higher levels of beta-carotene. Okay, so in this case we have the transgenic rice plant that expresses phytoene synthetase and CRT1 so that beta-carotene can be produced. Okay, and there we have it. Apologies for my pen. Okay, so I mean, this was not really meant to be a very technical discussion about how this happens, but it's just a discussion about why you would want to do it and essentially how you achieve this transgenic organism. So why would you want to produce a transgenic organism, and what does it actually mean to have a transgenic organism? It's expressing genes from other species. Okay? Right. Next, we will discuss how these processes are happening. So how we might identify and extract the gene in the first instance, how we might then insert it into a different organism, and then finally we might also discuss, or we need to discuss how we identify transgenic organisms. Alright guys, good luck.